Hello everyone. In this lecture, I will explain the development and composition of Periderm and then Rhytidome, dome, and Lenticels. The term periderm is collectively given to the protective tissue phloem and phloderm and the meristem that lies between them and gives rise to them. This meristem, the phelogen, similar to vascular cambium, is a uniserate layer of initials. Cells of phelogen is by periclinic division give of derivatives from the adaxial and abaxial faces. A phelogen produces pheloderm adaxially and phloem abaxially. Phloem, a major constituent of periderm, is generally subrized and inhibits translocation of water and solutes to tissue abaxial to it. As a result, these tissues senesces and die. Senescence and die. The cork tissue forms a protective layer of the tree after the epidermis dies, and it is said cork is generally formed in the stem and root of dicotyledons, which have a continuous and pronounced secondary thickenings. Cork is not formed in leaves, with the exception skills of winter buds of certain plants. Cork again is an important part of secondary tissue, which is termed periderm. Periderm is usually divided into three parts. The phelogen. The phelogen और कॉर्क कैम्बियम, इसको कॉर्क कैम्बियम भी बोलते हैं और फिलोजन भी, कॉर्क कैम्बियम, फिलोजन और कॉर्क कैम्बियम। द फिलिम और दूसरा है और कॉर्क, विच इज प्रोड्यूस सेंट्रीफ्यूगली बाय द फिलोजन। फिलोजन से ही प्रोड्यूस होता है फिलिम। द फिलोडर्म, लास्ट फिलोडर्म। फिलोडर्म, a parenchymatous tissue in some species and produced centripetally by the phylogen। centripetally अंदर साइड डेवलप होता है centripetally, और फिल्म सेंट्रीफ्यूगली बाय द फिलोजन, ये बाहर साइड डेवलप होता है कॉर्क। सेंट्रीफ्यूगल, ये यूज करना है, सेंट्रीफ्यूगल, सेंट्रीफ्यूगल, सेंट्रीपीटल, पीटल अंदर साइड, फ्यूगल फू, निमोनिक फू, फू बाहर फूकना, पीटल पी जाड़ा अंदर साइड, पीटल फ्यूगल, फ्यूगल मतलब बाहर, पीटल मतलब अंदर साइड, पीटल पी रहा है और फ्यूगल फूक रहा है बाहर। निमोनिक ट्रिक याद करने का, सेंट्रीफ्यूगल बाहर साइड, फिलोजेन का कॉर्डोकैम्बियम, जैसे ये है, ये बाहर का एपिडर्मिस है, ये सेंटर का पीथ है, ये फिलोजेन है। तो अंदर साइड जो सेल्स डेवलप होने लग रहा है और बाहर साइड डेवलप हो रहा है, ये जो ये है फिलोजेन कॉर्डोकैम्बियम। सेकेंडरी मेरिस्मेटिक टिश्यू से डेवलप होता है, डी डिफरेंशिएशन का प्रोसेस है। परमानेंट टिश्यू होता है लेकिन सेकेंडरी मेरिस्मेटिक में कन्वर्ट हो जाता है, डी कभी कभार ये सेकेंडरी ड्री में, मेरिस्मेटिक में। ये जो है ये कॉर्डोकैम्बियम है, फिलोजेन इसको बोलते हैं, फिलोजेन। ये वाला फिलोजेन, फिलोजेन जो अंदर साइड सेल डेवलप होने लग गया, सेल्स डेवलप हो रहा, वो सेंट्रीपिटल फिलोडर्म, डर्म। और जो बाहर साइड से डेवलप हो रहा, फिल्म। फिल्म और कॉर्क। बाहर साइड वाला कॉर्क, ये फिल्म। कॉर्क कैम्बियम। मिडल वाला कॉर्क कैम्बियम। स्ट्रक्चर ऑफ फिलोजन। स्ट्रक्चर ऑफ फिलोजन। स्ट्रक्चर ऑफ फिलोजन। Phloem is a lateral meristem consisting of a single layer of initial cells. The cells are uniform, rectangular. ये जो फिलोजन का सेल्स है, रेक्टेंगुलर है। लेटरल मेरिस्टम कंसिस्ट ऑफ सिंगल लेयर ऑफ इनिशियल सेल्स है। एक सिंगल लेयर का इनिशियल सेल्स, द सेल्स आर यूनिफॉर्म रेक्टेंगुलर शेप में रहता है। इन क्रॉस सेक्शन विथ देयर शॉर्टर एक्सिस इन द रेडियल डायरेक्शन। इन अ TLS transverse longitudinal section, they appear as a regular polygons. The protoplast of phylogen cells contains vacuoles of various sizes and may contain chloroplasts and tannins. Phylogen has no intercellular spaces except in the region of lenticels. लेंटिसल्स इसमें कुछ भी स्पेस नहीं रहता है, लेकिन जो रीजन लेंटिसल्स का रीजन उसमें कुछ स्पेसेस रहता है, क्योंकि रप्चर करके बाहर साइड ये हो जाता है, इसीलिए। द फिलोजन हैज डिस्टिंक्ट कंप्रेशन होगा। द फिलोजन हैज डिस्टिंक्ट पीरियड ऑफ एक्टिविटी एंड नॉन एक्टिविटी। द एक्टिविटी पीरियड में और may not coincide with that of the cambium. However, in some plants, two periods of philogen activity have been noticed in a single annual period of cambial activity. स्ट्रक्चर ऑफ फिलोजन हो गया। फिलोजन जन is single layer of initial cells. Cells are uniform, rectangular. In cross section. Cross section. Phylum. Phylum or cork arises from the abaxial derivatives of philogen. The cells of phylum or cork are usually polygonal or radially flattened. This is polygonal shape and radially flattened and divide tangentially in a cross section. Cells are devoid of intercellular spaces except in lenticular regions. The cells of phylum divide tangentially. Cork cells are dead. They may contain crystal containing cell. They may be sclerids or even non-subrized. In certain species, the cork cells' primary walls are subrized and non-contain a thick suberin layer interior to the primary wall called the suberin lamella. This substance, suberin, is highly impervious to gaseous water and resists the action of acid. This phenomenon of impregnation of walls with suberin is known as suberization. 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 A philoderm. We discussed cork cambium, phloem, phylum, and phloderm. Phloem, phylum, and phloderm. The phloderm cells are living cells with non-submerized walls. They are similar to the parenchyma cells of the cortex, but if the phloderm is multicellulaire, they are usually arranged in radial rows. In some plants of the phloderm have chloroplasts and are photosynthetic. Similar to similar to parenchyma cells of the cortex. Parenchyma cell of cortex. But if the phloem is multicellular, they are usually arranged in radial rows. In some plants, have chloroplast and are photosynthetic. Cells. Origin and development of periderm. The phloem may differentiate in a living epidermal collenchyma or a parenchyma cell. Just before the onset of meristematic activity, the cell loses the central vacuoles. The volume of cytoplasm increases and it undergo a pediclinal division. Following the first pediclinal division, two similar cells are formed, of which the inner ceases to divide further. The outer divides pediclinally. The outer cell differentiates into the cork cells, and the inner constitutes the phloem initial and continues to divide. The initial of phloem cells occasionally undergo anticlinal divisions to keep pace with the increase in the circumference of the cork cylinder. The number of phloem layers is usually greater than the number of phloderm layers. Number of phloem layers greater than the number of phloderm layers. If the first formed periderm remains on the axial organ for many years, the other layers of cork show cracks and are shed off. Thus, the thickness of cork on the plant remains constant. Bark. The first formed periderm may be replaced by newer periderm. When such replacement takes place, the last periderm is always produced inner to the earlier last formed one. The later produced periderm may have its origin in cortex, primary phloem, or even in secondary phloem. Generally, two types of formation of subsequent periderm distinguished. For plants where first formed periderm is developed in inner tissue, the lateral formed periderm usually form an entire cylinder similar to the first formed periderm. Such plants produce ring bark. Plants in which the first formed periderm originates in epidermis or outer layer of cortex, the later periderm developed in the form of scales or cells. The concave side of this cell scales is directed outward. Such plants produce scaly bark. Rhytidome. Rhytidome. Whenever a new peridome is formed inner to the one already present, the tissues exterior to it become cut off from the inner tissue. The nutrition and plant supply to them is cut off, and such cells eventually die. A hard outer crust develops out of such tissue. The crust increases in thickness due to the additional cork tissue is being produced from beneath. All such cork layers together with cortical film tissues exterior to the innermost phloem are termed as rhytidome, outer bark. All tissues extended to the vascular cambium are included in the term bark. The living part of the bark bank inside the rhytidome is often referred to as inner bark. Commercial cork. The source of commercial cork is curious sober, suber, curious suber, which occur naturally in the countries bordering the Mediterranean Sea. In this species, phylogeny arises in the epidermis. It may remain on the plant for an indefinite period of time. For commercial purpose, the first form periderm is removed when the tree is about 20 years old and about 40 centimeter in diameter. For exposed, cells of phylloderm and cortex dry out in die, and a new phylogeny is formed a few millimeters within the cortex. The subsequent phylogeny produces cork, cork more rapidly, and in about 6 years, the sufficient thickness to be of commercial value. The stripped cork shows a rough outer surface and a smooth inner surface. The cork is valuable because it is impervious to gases and liquid, non-reactive, and has strength, elasticity and lightness. It is used for insulation, soundproofing, and in the manufacture of sports goods. Cork is unparalleled as a material for making stoppers for wine and champagne bottles. Champagne bottles. We may like to know why commercial cork is to be cut in a particular plane. A cork is several centimeters thick, and then the lenticels remain active for a long time and result in the formation of cylinders of complementary tissues which extend from the phylogeny to the surface of the phylum. This complementary tissue forms the patches of dark brown crumbling tissue found in commercial cork. Because of the radial orientation of the tissues, bottle corks to be in a direction parallel to the surface of the trunk. This way, the cylindrical lenticels extends transversely through them. Seeds of cork from the tree are rarely more than 3 centimeters thick. Cork with a diameter greater than that cannot be obtained by cutting in the usual manner. Large cork are usually cut from sheets of ground and compressed cork or from multiple sheets composed of layers cemented together. These kinds of cork are of low quality. These are help because the cork is valuable because impervious it is impervious because it is impervious to gases and liquid, non-reactive, and has strength strength, elasticity, and lightness. Used for insulation, soundproofing, and manufacture of fracture of sports goods, and also for stoppers stoppers for wine and champagne pen bottles. And then lenticels. We must also know about lenticels. There are restricted area of relatively loosely arranged cells in the periderm. Lenticel pro- protrudes above the periderm because of their larger size and loosely arranged of numerous cells. This is continuity of intercellular species of lenticels with tissue in the axial organs. Lenticels are highly differentiated lens-shaped areas of periderm. They are usually found on stem and roots and appear on young branches or other organs as rough dark patches. Due to many intercellular spaces, lenticels have a loose structure, mostly scattered over the entire surface of stem. If we examine lenticel, is usually look like a convex lens, both internally and externally. Development and structure of lenticels. Development and structure structure of lenticels. Lenticels originate from localized region in phylloderm, phylogen, not phylloderm, in phylogen that become continuous with the non-lenticular phylogen. Lenticular phylogen has more intercellular species and produces derivatives at a higher state than do non-lenticular phylogen. The first formed lenticel generally appear below a stoma or group of stomata. Cells below the stomata begin to divide in different directions and the chlorophyll in them disappears so that a loose colorless tissue is formed. The cells that are derived from the divisions become more and more pellicrinate until the phylogen of the lenticels is formed. The cells which are derived from the division of the substomatal cells as well as those produced toward the extended by the phylogen of the lenticels are termed complementary cells. After division, the number of cells increases and masses of complementary cells are pushed out and rise above the surface of the organ. In the temperate regions, lenticels become closed by the end of autumn season by a closing layer, whereas in some plants, lenticels are formed relatively early in some plants. With the bark, in other they may remain active for several years. Thank you, everyone. Like, share, subscribe.